Chapter 9 is on the cell cycle. If you look at the rest of this packet, we're kind of all over the place. Like we're gonna do a part on cell communication that actually goes back into chapter 5, the last section of chapter 5, and then we're in like like one random section or like half of a section of chapter 36 or something. But chapter 9 is the main chapter for this unit. So the first part is on the cell cycle. And the cell cycle is like, you can think of it like the life cycle. It's like the life that a basic cell will go through. And basically, like every cell is going to have a lifespan, and every organism needs to grow and make new cells. And like when you think about what is it, we have like chemistry and the periodic table of elements, and it's like, well, what makes something living versus non-living? And there's a lot of different things that go into that, but a key feature of living things that most biologists agree on is their ability to make more of their own kind. Their ability to kind of divide and and make more of like themselves. So when we say the reproduction of cells, that is cell division, which is pretty straightforward for you at this point. So kind of the question we're trying to answer here is like, well, how do these dividing cells actually distribute their chromosomes? The chromosomes are, got your DNA. How exactly do we like distribute like those chromosomes from one cell into, when we say a daughter cell, it's not referring to like gender, it's just referring to like if you have like the parent cell when it divides it makes two daughter cells. And the daughter cells are coming from what would be like the parent cell. So it's just, I don't know why I didn't use son cells, I don't know. Like that's just kind of the wording that they've been using for like a really long time. Now, the reason why, what are the roles or the reasons why a cell will do cell division? Well, first off, if you're like a unicellular organism, you're only made up of one cell, this would be how you reproduce. Like, this would be like your version of like the birds and the bees, right? Like, for lack of a better, like that's all they do is just mitosis. Now, multicellular organisms, we'll have like a process called like meiosis, where you make like the sex cells and stuff. It gets a little more complicated once you get to multicellular. But keep in mind, if you're a unicellular organism, they just do mitosis. That's how they would make more of themselves. Now, there's three main reasons why an organism does cell division. The first would be for development, right? At one point, all of us were just like a single cell. And then that single cell would have to keep dividing and dividing to make the new organism. And as that developing embryo does cell division, you start getting the tissue layers, you start getting like more specialized cells. So mitosis can help us like move towards development. Another reason why we need to do cell division is to renew or replace. Renew and replace are kind of sort of similar to each other, similar words. But like every cell is going to have a lifespan. Not every cell is going to live forever. And so most of our cells will have to eventually replace them. So, and the time frame from some of these cells will depend. Like your skin cells, I think it's like every few days you got to replace those layers. Whereas like your neurons, you don't replace them at all. And why would that or like in the cells that can make up your like digestive tract, you replace those the most. And why would that be? Why do you think you'd want to replace? Why would we be replacing like our skin cells and like our digestive tract cells more than say even our muscle cells? Muscle cells we don't make more of. Aditya?
Your like skin cells are exposed to like more surrounding.
Yeah, like doing this, it might seem like it's like, man, that's how dangerous is that? I'm actually like ripping off like little tiny layers of cells. And you see this when you get a new pair of shoes, right? And you get like kind of that chafing, like that slight like wearing, and eventually over time you wear them the entire day, you might get like kind of a little like a sore spot forming. What's a consequence of like your brain? And when I say brain, I want to be more, because there are some brain cells that are like support cells that do divide, but like your neurons in your brain, they don't divide. And your muscle cells, they don't divide. What's a consequence of that? Yeah, Artem's good. Yeah, like a stroke, like you basically, you get like a blood vessel that bursts in the brain. Like if you kill a nerve cells, or somebody who like has like some accident that really damages their muscles, that's very permanent, right? There can be a little bit of the neurons can like repair themselves a little bit. And same with the muscle. There can be some amount of healing, but they can't just divide. Like you get a cut on your skin cells can just like divide and like repair that cut. So that's kind of an important difference to make note of. And that gets into repair. So another reason why we're going to do cell division is to repair. So if you get a cut, we want to like seal up that breakage and do cell division to repair that. Yeah. So anyways, cell division isn't all of the cell cycle. I'm going to show you like there's some other parts of the cell cycle, but it's like the main kind of it's like probably like you consider like they say the integral part. It's like kind of the main show that we focus on when we talk about the cell cycle. So here's kind of showing like asexual reproduction. So this would be like bacteria or an amoeba. Remember the amoeba sisters? This would be. Anybody know what this would be? An amoeba is classified as the different like kingdoms of life and stuff. Yeah, the protists. Yeah, so this would be like a protist. They only do mitosis, right? This would be like I was saying, like growth and development. So like here's your embryo. And like, that embryo needs to be a larger multicellular organism, so it's going to do mitosis to make new cell types. And tissue renewal, so like in your bone marrow, like it's where you make your red blood cells, you have a bunch of these basically stem cells that will just keep dividing to make your new red blood cells. All right, so now this is like officially 9.1. Whenever we're doing cell division, we're making genetically identical daughter cells. So if I have a skin cell and I got a cut in my skin, I got to repair that cut, my skin cells around that cut, they will like divide themselves, but each new cell is identical to the old cell. The consequence, and that's typically considered a good thing, right? Because you want all of your cells to be on the same page, right? Like you don't want to just keep changing the DNA because then it makes it harder for cells to communicate with one another. A consequence of that could also be if you get a mutation, like a change in the DNA, that keeps getting passed on to new cell lines. That's part of evolution, which we'll talk about later on. DNA is passed from generation of cells to the next. When they say remarkable fidelity is talking about like kind of accuracy, like trust. So in other words, we pass this DNA on to the next generation of cells without much error. When we get more into the details of DNA replication a little bit later on in the year, there is some room for errors to happen, but it's really very rare that we get an error in the DNA when we pass it from one cell to the next. Okay, when we talk about the DNA, if we're talking about all of the DNA in the cells, all of the chromosomes, that is the genome. Okay? When we talk about a genome, if you're talking about like a bacteria cell, your prokaryotic cell, their genome is just one single circular DNA molecule. That's something to keep in mind, not so much for this unit, but just even for the AP exam in general. Remember, prokaryotic cells have circular DNA. It's just this one circular chromosome. Whereas eukaryotic cells are going to have multiple chromosomes. And that chromosome number is going to vary. Like, humans, we get up to 46 chromosomes if you have, like, you include mom and dad. Other organisms can like have varieties of different numbers. And in DNA, we package DNA into chromosomes. So a chromosome is DNA plus proteins plus is that how you spell packed? That doesn't sound right. P-A-C-K-E-D, is that packed? Plus packed. So DNA plus proteins, I'll show it to you on the next slide, they typically will just call that chromatin. But then if you package that DNA even more tightly wound around those proteins, then you get into a chromosome. Okay? Because DNA, if you unwind DNA, I think they say it's about like 2 meters. So like, it's like taller than me, I'm pretty sure. It's like, I don't know how long 2 meters is exactly in feet and inches, but it's right around the size of like a human. So we got to fit that long of a DNA molecule into a single cell. So when I say like packed, like when we make, like that's very, very packed. And the proteins that we wrap DNA around, they're called histones. And you could think of like histones like, I always think of like a laptop charger. Where like if I unwind this laptop charger and it's really long, that would be like your chromosome. but you got to fit it inside your backpack. So you would wrap it around like these proteins that would be like maybe your like the power generator thing, I don't know, converter thing of your laptop charger. And this would be like one single chromosome, but you have 46 chromosomes in your cell. Right? So imagine if you have, like, what would happen if you had 46 laptop chargers that you didn't wrap around like this? What would happen? Yeah, did you? Probably not fit inside your backpack very well. But even more practically, if I'm trying to, cell division, we got to divide up those chromosomes. If I don't package them up kind of neatly and stuff, what would happen if I'm trying to remove those laptop chargers from the backpack and they're not tightly wound? Sean Connie? They get tangled, right? So that's another reason why we want to package up the DNA. It helps keep them organized, and those chromosomes won't get tangled up. All right, moving on to the next slide. Yeah, so here are your eukaryotic chromosomes. So the purple color, that would be the chromosomes, and those chromosomes then are wrapped around those histone proteins. Anybody want to guess what these pink streaks are? Trini, what do you think? No, the chromosomes would be like, this would be your nucleus, and the chromosomes would be all the purple stuff. But you see all those like pink, yeah, the pink streaks like coming out. It's the cytoskeleton. I think it's kind of neat. Anyways, all right, the structure of chromosomes. So this whole thing would be a chromosome. But this would be after the chromosome is duplicated. And this can be a very confusing concept, so I really want you to understand this. When you think of the classic chromosome, people tend to think of this, right? This kind of like X-shaped thing. Oh, and where the name comes from, some is referring to the Greek word soma, which is means body. So like, I don't know, like you can imagine like that kind of looks like a little body, like, like, and I think that's kind of where like that name. And then chromo, I don't know what chromo means. Oh, light. Chromo means light. I don't know really what that has to do with the chromosome, but I think it has to do with like them seeing it with a light microscope or something. But anyways, the soma, the body, this kind of looks like a little person. And this is the duplicated chromosome. So this is a chromosome after cell division. So if we haven't done, not after cell division, I'm sorry, after DNA synthesis, you've replicated the DNA, made a second copy. Meaning if you haven't duplicated the chromosome, this would be what the chromosome would look like. You would just have one, we call them sister chromatids. You would just have one sister. But then if you duplicate the chromosomes, then you get that second sister chromatid. Okay? And whenever DNA is duplicated, it's called 2N. N is referring to like a complete set of chromosomes. So like in humans, N is 23. We have 23 copies of chromosomes. So we'd have like 23 sisters, but then 2N is after you duplicate it. Then you get that second sister, so you get that second copy. Okay? Another little vocabulary here. Where we connect the sisters together is this center region where each sister has what's called a centromere. And I want to emphasize, because this is another common misconception. People tend to think there's one centromere connecting the two sisters. There's actually a centromere on each sister chromatid, and then there's proteins that will then wrap around and combine those two sisters together. Okay? And then another thing you probably didn't learn in freshman bio, but then all along the length of the two sisters, there are these, they call them cohesins. So IN, it's referring to a protein. There are these proteins that will keep them together along the arms. These are the arms. So that's an arm. That's an arm. Each sister chromatid has 2 arms. So there you go. Any questions about that? As I said, that you've seen something like this since like 7th grade, and it should be something that isn't like too mind blowing for you at this point. All right. Moving on. Yeah, a lot of this I've kind of already said. chromatin, that's DNA and proteins. Then if I keep packaging that DNA and proteins at even more and more packaging level, then you get down to the chromosome level. Every eukaryotic species has different numbers of chromosomes. So humans, we have 46 chromosomes, but it's 2 sets of 23. So that's what I was saying. The 46, they call that the 2N number, because you've done DNA replication, but there's only 23 different chromosomes. That would be your N. So like if you see this picture, this is showing the 2N level. This would be showing a cell that's done DNA replication. It would have 46 chromosomes, because you'd have 1, 2, you'd have 46 sister chromatids. But if that, like, say you get to the gametes, like the sperm and the eggs, they do something called meiosis, which we'll talk about in the next chapter, the next unit. They would only have 23 chromosomes. But then you combine the parents together, then you would get back to this level, where you'd have like 2 copies of each chromosome. Anyways, we'll get more into that stuff a little bit later on, but just kind of a little basic introduction to it. All right. The somatic cells, I told you soma means body. So these are the body cells. Like, in other words, these are every cell, all the cells except for the gametes, which are known as the sex cells. So like the sperm and the eggs. Okay? Okay. Yeah, so whenever we're not dividing the cell, the chromosomes won't be quite as packaged. They'll be a little bit longer, and they say a long, thin chromatin fiber. And why would that be? Why would we not just like keep the DNA super packaged whenever the cell is just doing its normal functioning? It's not dividing. Any thoughts? Like, why unpackage the DNA during like normal cell functioning and stuff? Think about what is the purpose of DNA. What are we doing with DNA? Aditya?
DNA still needed code for it.
Yeah, remember DNA has the information for proteins, so we have to then unpackage that DNA, when the the cell's not dividing, to still use that information to make proteins. Okay. If we're going to divide the cell, you have to then make a new copy of all the chromosomes. So you have to do DNA replication. And then you got to condense the chromosomes before we're going to divide the cell. Mention each duplicate chromosome has 2 sister chromatids. And then we combine those chromatids at the centromere. All right. So here would be like, here's one sister chromatid, right? So this is before cell division. We haven't divided the cell. Notice how it has a centromere. The centromere connects the 2 arms. Here's one arm. There's another arm of that chromosome. Then you do DNA replication, and then you get the 2 sister chromatids. So they're joined at their 2 centromeres, and then also they're joined along the arms by the cohesins. And that's what the process of combining the cohesins, that's chromatid cohesion. The cohesins are the proteins that are doing chromatid cohesion. That's holding the 2 arms together. All right, and then we do cell division, and we will then divide up the sister chromatids to your 2 new cells. And then, notice how then, like, here we went from N, then we doubled our amount. So now we're at 2N, and then we went back to our N number. Yeah, so this is just kind of in words what that slide was saying. Now, within cell division, yeah, what's up?
I'm kind of confused. So are chromatids just are the chromosome arms and the sister chromatids the same thing?
The chromatids are made of 2 arms. That'd be an arm, and that would be an arm. Yeah. All right. Within cell division, there's mitosis and cytokinesis. So mitosis, that is just when you divide up the DNA. That's just when you divide up the genetic material. So you're not actually dividing up the entire cell during mitosis. You're just dividing up. They also call this nuclear division. Mitosis is also called nuclear division because you're just dividing up the stuff inside the nucleus. Then cytokinesis, it's a one-step thing that happens like almost simultaneously with the endomitosis, where we actually divide up the cytoplasm. So cytoplasm. Okay. So gametes are produced by variation. Yeah, okay. So I mentioned that already. That when we make like the sperm and the egg, that's when we do meiosis. So it's going to be a separate chapter. But meiosis is almost just like you do mitosis 2 different times. And meiosis is going to give us non-identical daughter cells. So I said mitosis, you get the exact same cells, but then when we do meiosis, we get non-identical cells. And they're going to have half as much DNA as the parent cells. Okay, so like the parent cells would have 46 chromosomes, so they would be 2N. And in meiosis, you get half as many, so you just get 23, which is N. And then when you get like a sperm and an egg combining together, you would get 23 plus 23, or N plus N, and you get back to the 2N number. Okay. All right, let's take a little break before we go over the steps of mitosis.